Good morning. My name is Daniela Megrian, and I'm a research associate at the Bioinformatics unit at Instituto Pasteur de Montevideo. So, sorry, the slides are passing very slowly. I'm not sure why, but maybe it will take a bit longer. So, MoeA protein, it's an essential enzyme in all domains of life, so archaea, bacteria, and the eukaryotes. And it has an essential role in the biosynthesis of the molybdenum cofactor. So very briefly, the cells incorporate molybdenum inside the cell, and this molybdenum is inserted into a molecule of MPT by proteins MoeA and MoeA to generate the molybdenum cofactor. This cofactor is essential for the functioning of the molybdenum that catalyze redox reactions, as I mentioned, in all domains of life. But what's interesting about this protein MoeA is that it has acquired different additional functions during evolution. For example, in the animals, MoeA is fused to protein MoeA in the N-terminus, and this fused protein is called chefrin. This chefrin forms a mesh underneath the cytoplasmic membrane, and this mesh is used as an anchor for GABA and glycine receptors at the synapses. So chefrin is a moonlightning protein, so this means that the same molecule has very different functions in the same organism. So one of the functions is in the biosynthesis of the molybdenum cofactor, and the other one is in neurotransmission. And well, surprisingly, last year, our group identified a paralog of MoeA that we call GLP, that is exclusive to actinobacteria, and in this case, it has a very important role in cell division. So this protein GLP, very briefly, it's a duplication of MoeA that happened very early in the diversification of the actinobacteria. If we superimpose the structures of both MoeA and GLP, we see that they overlap extremely well, except for the angle between the domains that differs. If we look at the catalytically active MoeA, for example, the one present in E. coli, we see that the functional unit, it's a dimer that is symmetric, so it has two active sites indicated here. When we compare that structure with the dimer formed by GLP, this difference in the angle between the domains makes that the active site has a different structure. And this difference in the active site is probably associated with the loss of the enzymatic function. To try to confirm this, we map the protein sequence conservation on representative structures of the active sites of both MoeA and GLP. And we see that in GLP, the sequence conservation is very low, as it has very little pink compared to this one. So GLP is not a moonlightning protein in principle. It doesn't have the same molecule, two different functions, but instead, this duplication that happened in the actinobacteria led to one of the proteins maintaining the original function in the biosynthesis of the molybdenum cofactor, and the other one repurposing and acquiring an important role in cell division. So at this point, we were super interested about this versatility of this protein MoeA, and we wanted to explore what happened in all domains of life. So, doesn't change. So if we start with the prokaryotes, we see that the bacterial domain and the archaeal domain are mainly monophyletic, except for these two small bacterial clades that branch within archaea. So all canonical bacterial MoeA fall here in this big clade, and also GLP, so the actinobacterial one. If we look at archaea, we see that there was an ancestral duplication that led to the existence of these two paralogs, MoeA1 and MoeA2, that remained during billions of years of evolution. And if we look at the active site of both MoeA1 and MoeA2 of archaea, we see that both are very well conserved, so in principle, the catalytic activity is maintained. So this is a simplified view of this tree, where I mapped the domain organization of the protein MoeA. And we see that here in MoeA1 and also in the small bacterial clade associated to it, MoeA is fused to a PVP domain in the C-terminus. And if we look at the structure of this fused protein, we see that the PVP domain sits on top of the active site, probably channeling the substrate. So this PVP domain starts for periplasmic binding domain, And this domain belongs to a family of transporter of small molecules, like the MoeA system that I mentioned at the beginning, that allows the incorporation of molybdenum inside the cell. So we were revising the literature, and we saw that in some archaea it has been proposed that actually they don't have the molybdenum cofactor, but instead they have a tungsten cofactor that is a similar metal in terms of physicochemical properties. So we were wondering if this PVP domain is involved with the incorporation of tungsten in this specific parallel of archaea. To understand this better, we look for the co-occurrence of the fused protein MoeA PVP with all the membrane transporters known for molybdenum and tungsten. Because if the cell is creating a tungsten cofactor, first it has to incorporate tungsten inside the cell. So first we look for this in archaea, but we didn't find any correlation because basically most archaea or all archaea have all the transporters for molybdenum and tungsten. But if we focus on that small clade of bacteria that also has MoeA PVP, we saw a very clear co-occurrence between MoeA PVP and the type A transporter, that is a transporter that is specific for tungsten. So if we extrapolate this to archaea, these results suggest that this ancestral duplication of MoeA in archaea led to two different proteins that got specialized into a different substrate, one molybdenum and one tungsten. And this is super interesting to analyze in the context of the archaeal niches, because we know that tungsten is soluble in anoxic and sulfidic environments where molybdenum is not, and many archaeas live in such environments. And this is also very interesting to analyze in the context of the geological record, but I don't think I have much time to expand on this. So if we pass to the eukaryotic domain, this is a phylogeny of MoeA in all the domains of life, and we see that the eukaryotic MoeA has a bacterial origin. We found MoeA in almost all eukaryotes with a few exceptions. Notably, in fungi there are some absences, for example in Saccharomyces cerevisiae, the model organism, and we don't know exactly what this means. This is another view of the same phylogeny of the eukaryotes, where I map the domain organization of MoeA. As I mentioned at the beginning, in the animals, MoeA is fused to MoeA in the end terminus, but we saw that in algae, plants, and the SARS, so in some unicellular eukaryotes, this MoeA domain is actually fused in the C terminus. And this is very intriguing, and we don't know if it's associated with the moonlightning protein as well or not. But we wanted to know a bit more about this. So first of all, we analyzed the conservation of the active site of all MoeA proteins, and we saw that in Sheffrin, in the SAR, algae, and plant MoeA, the active site is very well conserved, as we saw in bacteria. So in principle, the catalytic function is maintained, but this still doesn't say anything about the probable moonlightning function in the SAR, algae, and plants. But we saw something very interesting in this other view of the protein. This is a monomer of MoeA, and we see this patch that is very well conserved in the SAR, algae, and plants. And very intriguing, this patch is also very well conserved in the animals. And if we see for example, this is the rat Sheffrin, we see that this spot is exactly where the GABA and the glycine receptors bind. So this makes sense because, well, this is an important region in the animals, but the SAR, algae, and plant, they don't have a nervous system, so we don't know what this patch is doing. As expected, in bacteria, this region is not conserved. So we're super curious about this. Our hypothesis is that probably this is binding an ancestral type of receptors that appear early in their eukaryotes, and we would love to validate this experimentally if someone works with SAR, algae, or plants. So, well, to sum up, in this work, we made an evolutionary analysis of MoeA over billions of years of evolution. First of all, we identified an ancestral duplication in archaea that led to the specialization of one protein into tungsten and another one into molybdenum. We identified a duplication within actinobacteria that, in this case, this actinobacteria maintained the two copies. One kept the original function, and the other one was repurposed into cell division. In the eukaryotes, we identified at least 2 independent fusions with the MoeA protein, one in the C-terminus and the other one in the N-terminus. And we saw that this fusion was an essential step for the acquisition of the moonlighting function and eventually the emergence of the nervous system. So if you are interested in the topic, we have this very nice article that we published on the history of GLP last year. And the overall story is currently in bioRxiv, and hopefully it will be published very soon. And before I finish, I wanted to thank the group of Pedro Alzari and Anne-Marie Benkel, because this work started when I was a postdoc at Institut Pasteur in Paris. The group of Rosario Duran at Institut Pasteur de Montevideo, that they were in charge of the proteomics for the identification of GLP in actinobacteria, and to the funding agencies. So, thank you.
Thank you very much, Daniela. Time for questions from the audience.
So you showed the conservation of the active site, which is always a really important thing, but there's always allosteric interactions, so other parts of the enzyme which are important for plasticity, motion of the enzyme. Did you look for any of those sort of secondary research sites? So I was wondering whether the binding of the glypeptide, is that an allosteric site, or is it that's then being hijacked, or do you think that's a completely evolved binding site?
You're talking about the active site or this other site that I show at the end?
So I'm saying not the active site, but there can be allosteric interactions within the enzyme that are other conserved sites outside. And I'm wondering whether that patch that became the binding site for the glypeptide protein, whether that's or the glypeptide, whether that's an allosteric site that's being hijacked, or whether it's something that's evolved completely separately.
Yeah, so the thing is that when we look at the conservation at the whole protein level in archaea and in bacteria, the only conserved region is the active site, nothing else. There is a glycine, if we have a view on the top, that is conserved in all domains of life. We don't know what it's doing, but it's very conserved everywhere, and we guess it has a functional implicance in the catalytic function. But then the only other region that we see conserving the protein appears in the eukaryotes, and it's that spot that I show. Otherwise, there is nothing else conserved on the structure.
Any other questions? I have one short one. Which approach did you use to find the autologous? And not which tool did you use?
Sorry, to find what?
Which approach did you use to find the autologous?
Well, actually, it's funny. The sequences are super similar in all domains of life. It's we started originally. So the first article was the one in GLP. So we started working with the orthologs identified in actinobacteria, and then with the HMM profile that we made, we used like Hammer for the identification in all domains of life, and it's super clear. There is no doubt that we are talking about MoeAs. Then we predicted all the structures, they overlap extremely well, so it was very easy to identify. It's not a difficult protein to work with at sequence level.
Any other questions?